All right, so thank you everybody for joining our first week of the micro course. It's really exciting to see the great turnout, which also means there's a huge interest for this course, which we could sense based on the responses that we got on the survey form. So on behalf of ICI Core, I would like to welcome everybody, not just the Emory members, but our affiliates from Georgia Research Alliance, as well as Georgia CTSA. So welcome everybody from the universities across Georgia. In this course, so we are part of an ICI team, which is led by our scientific director, Adam Marcus. Neil used to be our co-director. I still have him as in the picture because it's very still difficult to imagine not to have him. And I want to thank him for his support because this course was, the idea of this course really happened while he was here. Laura is our technical director now, and Stoyan, April, and I are the people who manage the microscopes along with Laura, and Stoyan is the main image analysis person in the group. So the ethos and the fundamental of ICI is to help you support with any of the microscopy experiment. We do consulting, and we also educate people. And this course series is part of the education initiative of ICI. As always, consultations are free. You can always reach out to us at ici@emory.edu and chat with us about your experiments, your protocols related to any imaging, your grants, as well as data analysis. Okay. At ICI, we have about 19 different microscopes that take care of different modalities: wide field, confocal, point scanning confocal, spring disk confocal. We have got light sheet, and so on and so forth. Okay, so if you're interested in any of the microscopy methods or techniques, please do let us know. You can visit ici.emory.edu to get idea about all the different offerings that we have. And that brings me to the agenda of the course today, which is why even we need this kind of course, right? So I'll be talking a bit more about that in the introduction part of the course. We will cover up what a perfect imaging experiment is. Now, ICI did micro talks where April, me, Stoyan, Neil, and Laura have mentioned a lot about what an imaging experiment is, things and nooks and crannies about how to do an experiment, how to figure out image settings, how to do a bit of image analysis over there. And I'll kind of try to cram all of that in a short time so that we are all up to speed with this. And later on, towards the end, I'll put a fundamental or a base of what an image is. And that is going to be helpful for the few coming weeks that we will be talking about pretty much image analysis. Okay, so why do we need the course such as this, right? So we always get feedback from users, and one of the biggest feedback that we get is, well, we have a lot of data. What do we do with that, right? And what happens most of the times is this kind of discussion happens towards the end of the imaging session. So we thought we really need to have a course in front where you can think about the aspects of image analysis, and hence the planning of the experiments that you should think about, not just at the bench, but also when you come onto a microscope, right? So at the end of the day, everything starts with what is your question that you are trying to address. And once you know what question you are trying to address, we can build on top of that, be it in terms of planning the experiment, be it in terms of using a particular kind of microscope as well as a microscopy method, as well as thinking about the image analysis right from get-go. And that's something that brings to us this kind of a course. Apart from this, we also get users who tell us, oh, well, I saw this particular kind of an image in the paper, and I want to have a figure that looks like this, right? So for example, here, the authors are trying to quantify the talpid 3 fluorescence intensity, and they are quantifying this based on each of these yellow circles that you are looking at right now. In your images, you don't want to go and circle your region of interest manually, because that can be a lot of work when you are trying to compare things between your wild type cells versus your mutant or knockout cell types, right? So you want to have an automated image analysis workflow, and that's where a course like this is going to be very helpful to you. Apart from that, the core has different microscopes that will let you do or let you image multi-well plates, right? Which means if you have a 96-well plate, you can have cells with different treatment types or different knockouts. And how do you quantify data, right? That's the biggest hurdle at the end of imaging a 96-well plate. What do you do with that data? How do you quantify and analyze all of that data? And this is exactly where some of the tools that we'll be teaching you in this course will be very helpful to you. Now, sometimes, you take images and it's not just about data analysis. Sometimes the images are qualitative in nature, which is you did your Western blots and you are like, okay, I just want a couple of pretty pictures that represent what I'm seeing in the Western blot, right? So this course will also help you with better image representation, better image reconstructions, and so on. And if you wish to, you can also extract some of the features that you are seeing in this images. So this course will help you with literally everything that you can think of with your image analysis data. Now, we have a huge diversity of attendance at this course. By and large, 80% of you who are attending this course are beginner, right? And that is really wonderful, which also means we will go really very slow in this course. Now, this course is not just a bunch of lectures that we are trying to throw onto you. This course is going to be a mix of lectures and workshops. Today, it's just an introductory lecture. So, you can let us know, you can give us your feedback. You can let us know what is it that you want to learn apart from what we have outlined in the course. So it's really important for us to get your feedback and not just throw things onto you, right? So 80% of you are at the beginner level. A lot of you have got ongoing imaging projects, right? So from the names I saw earlier, a lot of names are not familiar to me based on the survey that some of you took. So I want to pause here for a minute and want to make sure that everybody who is present right now here has filled out the survey form. Now, why is that important for us? It's important for us because this is how we will communicate with you about course announcements in future. Let's say if I need to tell you that, oh, I've posted something, have a look at that. I'll be messaging you through the emails, right? So I want to pause here for a minute so that you can complete the survey form. April or Stoyan, they're gonna
I already posted it as the first thing.
So April has posted the link for the short survey. Please have a look at that and complete the survey form. All right, so I'm gonna shut my mouth for a minute.
I want to add that I also put up the links for our Slack channel as well as our micro talks.
Thanks, April.
Yeah.
And as you get done with this, if you could just give a thumbs up or any weird emojis that you want to use, that would be great. Does anybody need any more time, or should I just move on? I know this is one of that boring part of the courses where you have to do things when somebody tells you to do. Okay, I think I'm gonna move on. It's a very short survey form, which you can fill while I'm chatting a little bit here. Okay. All right. So moving on, um, what is it that we intend to empower you with, right? And I'm gonna use this word, which is empowering you with tools that help you go from a vision that you have about data towards executing that vision. All right. So first, we are gonna teach you to do things using Fiji. Fiji stands for Fiji is just ImageJ, literally. Okay, and what can it do? It can do literally everything that you can think of, and it does so using thousands of plugins which Fiji comes with. So if you look into the frequently asked question tab of our website, you will find a download link for Fiji. And if you have any issues download with the downloads, and once you have downloaded Fiji, do let us know, because from next week we will be using Fiji to do a lot of things. Okay. Now, um, what does something like plugin mean, right? So think about Fiji as one of those coffee makers in which you put on a pod, right? So if you want to drink a coffee with, let's say, a nutmeg flavor, which is a very terrible flavor as a matter of fact. But if you wish like drinking something like that, what you do is you get this coffee pot, you put that into the coffee machine, and boom, you get a coffee, right? So Fiji as a backbone is like that coffee machine, and each of this plugin is like that pod that goes in and gives you the coffee that you want to drink. So in nutshell, using Fiji, you'll be drinking a ton of Java, and pun intended, Fiji is based on Java, and hence this corny joke of Java really fits in. Fiji can open literally any image file format. So if you are imaging, let's say on Leica or a Nikon or an Olympus, you can literally drag and drop those files into Fiji, and you should be able to open them. You always wonder, oh, I want to image my nucleus, which looks like blue. But again, these colors are just pseudo colors, right? So you can image things the way you want to. At the end of the day, everything is grayscale. You can bring your images in Fiji and apply a particular kind of lookup table and make it look the way you want to. Okay. So we will be looking a lot more about that. We will be looking at how to generate composite images, addition of scale bars. These are really basic things. If you have a three-dimension data set with lots of Z sections in them, right? If you want to generate maximum projection, we will look at different plugins that let you do nice maximum projection constructions. You can also generate very basic volume reconstructions of your 3D data set, and also assign sort of a lookup table which gives you different colors for different Z planes for your images. Okay. And then comes what's really this course is about, which is image segmentation. And right now, if you think, I don't know what he's talking about in terms of image segmentation, it's okay. As we keep on moving forward, all these things will become very clear, because we will be doing workshops. And the way the workshops will be held will be, I'll show you something for 5 minutes. You will get time to execute that. I'll again show you something for 5 minutes. You will again get time to execute this, right? So this will be sort of an interactive workshops. And if you don't have images with you, don't worry. I'll be posting everything on our GitHub page, which I'll talk about in a bit, okay? So coming back to segmentation, right? So let's say, for example, you get your nuclei over here, and you want to identify or measure the fluorescence within this nuclei. Each of your image will have certain intensity values, and that's what you are looking in this section right now. And you can select what intensity values you want to measure and create sort of a mask out of it, right? So I want to select image intensity values which are between, let's say, 6,000 to about 65,000, right? That's what I'm selecting over here, and that's what this gray area is. Or if you want to simply select the nuclear membrane, and there are many different ways to select nuclear membrane. And this is just one of it that I'm showing you over here. So you can select intensities which are roughly from 3000 to 6000 for this image. And then if you create a mask, boom, now you get an area which enables you to select just the membrane. Okay, so this is where image thresholding and segmentation in Fiji will come very handy. Apart from that, images are nothing else but pixels, and pixels are nothing else but numbers. So when you think about that, in terms of when you think about images in terms of numbers, you can do a ton of things with numbers, right? You can do a lot of math. You can use a lot of filters, which are nothing else but execution of bunch of scripts, which has to do with math, and eventually do something like binarization in terms of generating a mask, which you just saw earlier. You can also automate the workflow. We will be focusing a lot, not on automation, but a lot on doing things. And once you understand how to do things, trying to fit everything you are doing into a micro language will become very easy. And Stoyan is going to hold a workshop on how to work with micro languages. Okay, and this is in terms of how to automate your workflow. Apart from that, there are a lot of models we'll be using. Let's say, for example, StarDist. So you all have heard, and I probably fancied by the word deep learning, right? Everything is so much about deep learning and artificial intelligence these days. There are plugins which integrate along with Fiji. So one of the plugins that integrates into Fiji is called StarDist, and StarDist is based on UNet, which is type of a convolutional neural network based deep learning model. And in this particular plugin, the model has been trained based on a ton of different nuclei that people annotated. And again, if the word annotation is new for you, that's okay. We will be getting into annotation in our workshops later on, okay? So what essentially happens is people train a lot of nuclei, which are very different, and that's get fed into this model. So now when you are using a particular image with very complex nuclear shapes, you are able to segment the nuclei. So for example, early on here, this nuclei are very easy to be segmented using Fiji because they are spatially separated from one another. But if you look at this nuclei, for example, if you are using tissues, right? These nuclei are very connected to one another, and it becomes a problem to segment them using Fiji. And this is exactly where deep learning models such as StarDist come into picture. Now, StarDist can also be trained using zero-cost deep learning Google Colab notebooks as well as Python. Now, these words might be scary for you. We won't be getting too much into Python in this course, but we will try to expose you with Google Colab notebooks. And this is just our first version. In the second version of the course next year, we will come up with workshops where you can do much more interaction with Google Colab if you don't feel comfortable doing it by yourself. Apart from that, so I just talked to you about StarDist, which can deal with nuclei, right? How about something like cytoplasm? So with regards to cytoplasm, and pardon my annotations over here, this data set is also courtesy of Dr. JP Stevens and Dr. Vasanta Kolachala from Nikita Gupta's lab, where they are trying to segment cytoplasm within the liver, right? So it's very difficult to identify cells over here, but using Cellpose, which is also a deep learning based tool, you are able to identify both nuclei as well as the cells very easily, which otherwise, without an appropriate label for hepatocytes, it would be very difficult. Cellpose is a standalone tool, and we will send you the links for downloading Cellpose as and when we use this in the workshop. So it's a standalone software or a tool, and it has got a very easy graphical user interface to work with, right? So you can upload your image into the tool, and boom, with few simple clicks, you get segmented data sets, right? And you can import the segmented data sets into Fiji. You can identify the mean fluorescent intensity per cell, not just the entire image that you are throwing, but individual cells, okay? So we will be using Fiji, StarDist, Cellpose, and what else? We will be using Cell Profiler, right? So I talked about automating your workflow. Now, Cell Profiler also helps you to analyze your images. It's really good with your two-dimensional data sets. Well, it also does three-dimensional data sets, but if you have cells which are plated on a dish and you are imaging that, it does a really good job with that. Cell Profiler is very modular, which means you need to understand what each of those modules are doing. And hence, using or learning Fiji really comes handy. Fiji comes handy because when you want to do something, let's say I want to look at my image histogram, or I want to adjust brightness contrast to my image, or I want to do something like median filtering with my image, Fiji really gives you an output right away. Whereas with Cell Profiler or any other tool from different softwares for image analysis, you have to play with it. You need to understand what that's happening, okay? So let's say, for example, in terms of Cell Profiler, you have nuclei and you want to segment something outside. You can use a bunch of these modules. But again, as I said earlier, and I want to re-emphasize, you need to know what each of these modules do, and hence learning Fiji is the right way to go before we reach to the Cell Profiler stage, okay? Apart from that, we will also be looking at different tools which can help you with 3D reconstruction. Now, these are not free tools. These are the tools which are part of the workstations on core. The reason why we will emphasize on these tools is because they are really very easy to use, and they are really very powerful in terms of 3D reconstructions, giving you really awesome data. So when it comes to publishing papers, we strongly recommend you to try and use some of these tools versus some of the freely available tools available in or plugins available in Fiji, okay? So once again, I want to reemphasize Imaris, Arivis, we have licenses for both of them, and they're extremely powerful for 3D reconstruction, 3D image analysis, and so on. Apart from that, we will also be learning something called colocalization, which is a big topic. So Stoyan will be spending one workshop simply on colocalization analysis. And we will end the workshop with image classification and segmentation, right? So if you are watching a show on Netflix, you always want to leave the season on a cliffhanger. This is kind of us leaving the season on a cliffhanger, which is boom. You learned image classification and segmentation. You learned how to annotate a bunch of images. What do you do with that now, right? So we will reconvene sometime next year, where we will be doing a lot of deep learning based tools, and all the image annotations that you will be learning in this season that will be really applicable to what we will show to you in the next season, okay? And is it just me and Stoyan who are gonna just keep on talking? Absolutely not. We also have a bunch of guest lectures. We have, and these two folks are really terrific with what they do. We have Dr. Beth Cimini. She is a group leader at Broad Institute. She is one of the member who is part of the developer team for Cell Profiler. So in the first part of Cell Profiler, I will be teaching you stuff. And if you have more detailed in-depth question, Dr. Cimini is the right person to answer a lot of those questions. She is also part of the Center for Open Bioimage Analysis, which does a lot of workshops on Cell Profiler. We also have Dr. Sreenivas Bhattiprolu. He is head of digital solutions at Zeiss Microscopy. And he's also part of one of the open source tool called Apier, where you can use a bunch of modules to do image analysis. Apart from Apier, Dr. Bhattiprolu, he also has a channel on YouTube called Digital Srini. I'm a huge fan of him. I follow his videos all the time, where he ends up teaching people Python to do image analysis. So if you're interested in learning Python in relation to image analysis, I strongly recommend going and checking out his channel. This is one of the best channels you will find to learn Python and learn image analysis. Okay. Apart from that, some of the housekeeping stuff. Slack. Slack is the currency of us communicating with each other. During the course, if you have any question, we will be answering that to you based on Slack. Okay, so please join our channel. I'm again going to pause for a minute. April has posted the links for our Slack channel. Please go ahead and try to log in. If you have any questions, this is the time. Let us know. Send us your questions on Zoom chat. We will try to help you with that. Okay. So I'm going to pause here for a minute, and please go ahead and join our Slack channel. Okay. So I'm just going to pause here for a moment. If you have any questions about joining Slack, reach out to us. If you think that you cannot join Slack, send me your email. I'll send you an invite, and that will make your life much more easy. So I'm going to take a pause for a minute. Okay. Apart from the Slack that I talked about, I will be posting the course materials on the GitHub link that April has posted. Every week from next week itself, we will be doing much more interactive sessions, right? So when I say I'll be showing you something for 5 minutes, we want to work on the same images, so that those images will be on the GitHub. Right now it looks very empty, but as time progresses, I'll be posting everything, let's say by noon or every Wednesday. Okay, so if you go on the GitHub page by noon on Wednesday, you should be able to find everything in a very systematic manner cataloged on the GitHub link. Okay, so that's what we'll be using. And feel free to email us at ic@emory.edu if you have any questions on anything. Okay, so switching some gears, what is a perfect imaging experiment, right? So I mentioned earlier that it's great that we want to do bioimage analysis, but it is really limited based on the experiment that you are doing. Okay, so what is a perfect imaging experiment? The first step of any imaging experiment is planning and performing the experiment on your bench, right? And this also means you'll be discussing things with your lab members, you'll be discussing this with your PI and so on. The second step is taking an image. And in between planning, performing the experiment, as well as taking an image, comes us, right? So you can come to us, consult, we will help you with things. And imaging really means you are choosing the right microscopes with the final goal, which is data analysis in mind to acquire your images. And last part is analysis and image quantification, which is the main focus of this course, right? And in between imaging and analysis is how you store your data. There are a lot of different things about data storage. We won't be talking much about you, but at ICI, we have a great structure in terms of cloud where the data stays for 2 years, which is just fantastic, okay? So an intersection of planning and performing the experiment, imaging and data analysis is where a perfect quantitative imaging experiment resides, right? So a perfect quantitative imaging experiment is not just about you coming and imaging and using the tools, but it's at the intersection of all these 3 different things. Now, we talked about all of this, planning, performing, and imaging in details in our microtalk series. So if you are interested in going back and revisiting in detail what any of these things are, it's on our microtalks version 2 webpage. We will be updating some of the links to for you to go back and watch the recordings. So please do take advantage of the detailed talks. What I'll be doing today is kind of summarizing and cramming everything that April, Neil, Stoyan, me, and Laura went through. Laura's talk is still left, so I want you all to, if you're interested in understanding how to put all these images that you have taken into figures, methods, and executing that towards publication stage, Laura's talk is going to be this Friday at 1 p.m. and the link will be on the website that April will post. So please go ahead and join us on Friday at 1 p.m. for the last talk of our micro talk series. So going back to planning and performing, right? So the planning and performing part really starts with what's the goal of your study. So if you have a particular question, let's say, for example, what's the effect of a drug on cell morphology or protein distribution between cytoplasm and nucleus, or let's say, for example, effect of a drug on cell viability, you will try to accumulate different strategy. You will talk to your lab members, talk to your PI, and be like, okay, I can use a certain kind of a chemical probe, you want to order that, or you will have your cell type expressing a particular fluorescent protein, or you will use an immunofluorescence strategy, right? So you will use either of these strategies to come up with an experiment plan, and you are going to execute your plan. So let's take, for example, each of these questions and try to divide into a possible scenarios that might arise when you are thinking about an experiment or even you are executing that experiment on the microscope, okay? So again, if your goal is to quantify the data, it is really essential to prep your sample as well as to acquire data in a manner that facilitates image processing or analysis downstream of everything that you have done. So let's say, for example, your goal is to understand the effect of a drug on cell morphology or distribution of the protein between cytoplasm and the nucleus. Okay, so you do this experiment, which is, for example, immunostaining experiment. You fix your cells, you image, and now comes the big part, which is, okay, I want to measure oops, hold on a second. I want to measure the area of my cell, okay? Or the intensity of the protein in the nucleus or the intensity of the protein in the cytoplasm. So what would you do? I showed to you image thresholding and segmentation, right? So we will take advantage of image thresholding and segmentation that you are seeing in this pipeline. You can do some image math and say, subtract the nuclei area from my cellular area, which will now give you area of your cytoplasm, okay? And it's really important when you are planning your experiment to incorporate labels such as actin or a cytoplasmic dye or some kind of a cell membrane label, because all the tools that we will be looking at, they rely on these labels to achieve segmentation, okay? So that's the first important step when you're planning an experiment. Now, having said that, we can use some of the deep learning tools, right? Because you will be like, oh, well, you just showed that data from liver from Nikita Gupta's lab where you told us that, well, there was no label, but some of the deep learning tools were still able to identify the cells. Well, that's true, but we want to make life of these tools easy. An alternate way of still making the lives of these tools easy is if you are using the autofluorescence as a readout. But again, things like autofluorescence and so on can be very dirty. When I say dirty, it means the signal to noise ratio is really terrible, right? So I'm throwing one more technical word here on your head, which is signal to noise ratio. Imaging at the end of the day is about signal to noise ratio, right? So if your signal to noise ratio is really good, everything is very easy, all right? So let's take an example, which is effect of a drug on cell viability, right? So when we are talking about effect of a drug on cell viability, you would like to know whether my cells are live or dead. And there are different probes which are available to investigate this. For example, propydium iodide, annexin-5, or even caspase-3, which specifically helps you investigating apoptosis. Now, when you are using either of these probes, what's going to happen? What's going to happen is your fluorescence is going to go from low to high, right? So if you bring your sample, your cells, you put them on the microscope, and you are like, okay, I'm going to set my imaging parameters, and I'm going to get some signal. There's going to be a problem. What is that problem going to be? Because you're going from something like this on the left to something like this on the right, where something is fluorescent. Most likely you will end up facing a problem where your images will either be super saturated or your images will be undersaturated. Okay, and we will talk a bit more about what saturation means later on in this talk. Hence, it's important to select a control, for example, a positive control, which will help you decide your imaging parameters to figure out what your highs and lows will be. And your images won't be saturated, and you will be using an entire dynamic range available in your image. Okay, so again, one more big word, dynamic range, and we will be talking about that too later. Again, it's important to know what magnification and resolution you want to use, right? So for example, if your goal is to simply know how many of my cells are PI positive or caspase-3 positive, you don't need to go and use a higher magnification objective, for example, 60x or so on. Here you can use a low mag objective, for example, 20x or 10x, right? Because all you want to know is whether or not your cell is positive or negative for a particular probe. However, if you want to then go and look for where these probes are residing, now propydium iodide or caspase or annexin-5, they don't go into a particular part of a cell, so that's not a question over here. But if you are using any kind of a probe where you want to know whether they are part of a particular organelle, then you need to use a high mag and high resolution objective, okay? And that's where something like mitochondria comes into picture, right? So if you want to know the cell viability based off of mitochondria, right? Because when the cells are not happy, mitochondria do not have higher membrane potential. So if you're using a dye such as TMRE, which helps you understand mitochondrial membrane potential, what do you do? Or if you want to look at morphology of the mitochondria, what do you do, right? So in this situation, the fluorescence is going from high to low, and the cells are happy. They fluoresce really well with TMRE, and you are going to adjust your imaging parameters based on this high fluorescence level. So here you don't need to have crazy controls, because you're gonna set your dynamic range based off of this, and then you are going to go down in terms of your fluorescence levels as the cells are going to get unhappy, right? So as the cells go unhappy, they lose their membrane potential and the fluorescence decreases. So here, if you want to try to determine how an individual mitochondria is losing the membrane potential, you do want to use a higher magnification objective and higher resolution objective. But if you only want to look at a bunch of cells and see what membrane potential is doing per cell, then you can use a low mag objective. Okay, so these are a bunch of words. How does it look in an image? So here on the left, we have cells with mitochondria as label. Now you cannot really distinguish individual mitochondria on the left for objectives for images taken with a 20x.4 NA objective, right? And this is a wide field image. So this kind of an image is really good if you simply want to know what is happening with the mitochondrial membrane potential at a cellular level. However, if you want to dig into detail for individual mitochondria, you either need to use a 60x 1.3 NA objective, or you can go and do super res imaging to know what's happening dynamically to individual mitochondria, whether they are fragmenting, whether they are fusing with each other, what is the size of the mitochondria, and so on, right? So if you are doing drug studies where you want to know what's the effect on the morphology of the mitochondria, planning your experiment in advance can really help you, okay? Then some of the experiments that people publish, for example, something like DNA damage, right? So if you are using drugs, cancer treating drugs, and you are adding them to your cells and you want to know what's happening with my DNA, is there any kind of DNA damage happening? One of the most common immunofluorescent marker is gamma H2X. And this is a really classic comparison. If you are using even a high res objective, for example, 100x, and if you look at this little white structure with wide field microscopy, and if you look at the same structure with super resolution storm microscopy, what you will appreciate is that this appears as one dot to you, whereas that one dot, when you image through super resolution, there is still enough spatial resolution to know individual structures within this dot, right? So this is where you need to think holistically, what is it that I want to represent? There are people who use 40x magnification objective, and they still try to address the question, how many gamma H2X foci that they image? And that's not really right, okay? So if your drug is doing extensive damage to the nuclei, and depending on how the DNA damage machinery is responding, the number of gamma H2X foci may vary, right? So all of these questions depends on your cell type, depending on how much damage you are getting. And then you can think of what kind of a microscope I should be using based on my questions, okay? So that's something in terms of planning and performing. Then comes what kind of microscope that I'm using. And I did try to touch up on what kind of microscope to use earlier, because everything is really connected with one another. Here, I'll talk a bit more about image settings such as bit depth, aspect ratios, and in general, what happens in terms of input and output, and what happens in terms of imaging modalities that you are using, right? So there is no particular imaging modality that answers all your questions. There is always going to be some negotiation that you need to do when it comes to individual methods. So let's say, for example, if you are using a wide field microscope, right? So I'm gonna pull up all of them at the same time, where we are comparing wide field, the point scanning confocal, and the spin disc confocal. And we are looking at different parameters over here, whether the microscopy method is gentle, whether it's fast, whether it's easy to perform, how many colors you can do, whether it's isotropic. What isotropic means is you are getting the same resolution in X, Y, and Z dimensions, okay? How good of a resolution you can get. And resolution, again, depends on what kind of an objective you're using, right? So if you are using a wide field epifluorescence microscopy method versus point scanning confocal, these methods won't change the resolution. At the end of the day, resolution is dependent on the objective that you are using, whether you can do a large volume, whether you can do 3D, right? So let's compare each of them. For example, if you are using
Hey, Grav, I think I muted you on accident. Can you unmute yourself? Sorry. I'm so sorry. I think here.
I think, can you hear me now?
Yeah, I'm so sorry. I did that because I heard a bunch of background
that's
noise and I
okay.
muted
That's
people.
okay.
So I'm so sorry.
No problem. All right, so let me go back and talk a bit about comparison between different microscopy methods, right? So I'll just rehash. I don't know at what point I got muted, but let me rehash. Different microscopy methods can be broken down based on whether they are gentle, they are fast, they are easy, how many colors you can do, whether they are isotropic. What isotropic means is whether you are getting same resolution in X, Y, and Z dimensions. What kind of resolution you can get. Again, resolution is independent of wide field epifluorescence or point scanning. It's dependent on the objective that you are using and the numerical aperture of the objective. Whether you want to do large volume, whether you want to do a whole lot of 3D sectioning with your microscopy method, right? So if we compare, let's say, 3D part between wide field versus confocal methods, both point scanning confocal and swing disc confocal are good at doing 3D, whereas epifluorescence is really not good at doing 3D, right? If you look in terms of resolution, right, this is the point I was trying to make, which is resolution is dependent on the objective and not based on the microscopy methods. If you are doing multicolor, point scanning confocal is much more versatile in terms of doing multicolor. In terms of fast, if you are dealing with live cell samples, right, you want things to be fast and gentle. And that's where spinning disk and wide field comes into picture, whereas point scanning is not that fast. And hence, it's not going to be much gentle on your sample, right? So this schematic can kind of give you an idea as to what method to approach. And at the same time, reach out to us. This is why we are here. We really want to make sure that you are using the right methods and the right microscopes for your applications, and we can talk about that. Apart from that, no matter what microscope you use, there is something in, there is something out, right? In is going to be the lasers that you are using, and either laser or a light source that you are using if you're doing, let's say, fluorescence. So in terms of laser, what is it that you need to think about, right? You need to think about whether or not the intensity of the laser, the power of the laser, is that going to damage your sample? Now, photo bleaching is going to be a big deal if you are using live cells, but if you are using immunofluorescence, photo bleaching might not be a big deal because of the mounting media that you are using, right? A lot of mounting media come with agents or reagents that can quench the toxic ROSs that gets produced to bleach your fluorophore, okay? So if you are using immunofluorescence, photo bleaching might not be a huge deal, but definitely it's a big problem if you are using live cell samples. Scanning parameters, right? If you are using confocals, you need to think of what kind of aspect ratios I want, whether I want a 512 by 512, whether I want a 1024 by 1024, what kind of linear averaging I'm doing, right? All of this will determine how much exposure your sample is getting to the laser. And again, the speed and averaging. So all of this will matter with your photo bleaching. Apart from that, the channel setups, whether or not how many fluorophores you have, any kind of bleed through going on. If you can image things simultaneously, your sample will get less exposure to the laser. But if you need to do sequential imaging, there is going to be a greater exposure to the laser, and hence the chances of photo bleaching increases, right? And all these setups really are important if your fluorophore is dim or it's weak, right? So if you want to achieve higher signal to noise ratio at times, you will have to average your sample out so that you can remove much of that noise even before you acquire the data. And a good image is really important before you start image analysis. If you have lots of background noise, that's going to affect image analysis. And we will talk about some of the ways you can filter out this background noise. For example, you can use median filter to remove some of the noise that you get. But image analysis should not be a solution to remove the noise. The noise should be removed when you are taking the image, okay? So try to make your life easy when you take your images versus trying to use a bunch of filters to remove the noise. And again, the same thing if you are using lamps, mercury lamps, or LED filter sets, try to use the right kind of filter sets and be gentle to your cells as much as possible. Okay. At the exposure time will also increase the noise that you get and the background that you get. So be aware of that part when you are doing wide field rapid fluorescence microscopy. All of this also when you are trying to set up your imaging parameters, the gain, the detector saturation, the offset values, they also make a lot of difference in terms of the signal that you are getting, right? So this is kind of a whirlwind tour of what's happening when you are sitting on the microscope. So if you have any questions about this, we can discuss about it. Or when you are on the microscope, reach out to Laura, reach out to April, ask me about these questions. Okay, and we will help you walk over all these parameters and refresh your understandings. Okay. Few other things, signal to noise ratio, right? Everything is about signal to noise ratio at the end of the day, which I talked about a bit early. File, always save your images as raw data sets. And what do we mean by raw data set? What I mean by raw data set is if you are on Leica, save them as the Leica file, the LIF file. If you are on Nikon, save your data set as .nd2 files, right? And not just as TIFF file. And why should you save things as .nd2 files or .lif files and so on? That's because all these files also integrate the metadata along with the images, right? So the metadata is crucial at the end of the day. It helps you understand what kind of imaging parameters you had used. You can always convert your ND2 or LIF file into a TIFF file using Fiji. So don't worry about TIFF file, and never ever use JPEG as a way to save your files, right? Stoyan touched upon this on last Friday, that you should always use lossy format, which is .tiff format and or lossless format. But if you use something such as JPEG, you lose a lot of your data. So don't use JPEG format, use lossless format such as TIFF for everything you do with image analysis. All right. So moving forward and switching some gears, what is an image, right? So if we look at this mitochondrial image, and if we zoom into this small area, what do we see? Images get pixelated, right? So images are a bunch of pixels. However, what are these pixels? These pixels are a bunch of numbers. So in this example, if I zoom into a nucleus, and if I transform this in Fiji, what I see are a bunch of numbers. Everything which is dark or black over here are represented by zeros. Everything which is white or grays, I would call over here, has different numbers, right? And why is it that these numbers only go up till 200s? And the answer lies right here, okay? Where you see something called 8-bit, right? So the answer lies right here, which is it's an 8-bit image. And an 8-bit image has a dynamic range extending from 0 to 256. So everything, all the numbers are going to be in between 0 and 256. So the way I like to put that is you have 0 as your black, and you get different shades of grays over here. And the shades of grays go from 0 to 256 in the values, okay? So if this is a bit confusing, let's go further and try to understand a bit more about this. Because images and pixels are numbers, we can do a ton of things with these numbers, right? So we can do a lot of mathematical operations. We can use a lot of filters, which I mentioned a bit earlier. And these filters execute a bunch of kernels, which are again mathematical operations, and we can binarize each of these images to get something like a binary mask, which allows you or helps you in the process of segmentation. Okay, and we will be touching upon each of these things in details during the workshops. Okay, so where do these numbers come from? So I just mentioned in the earlier slide bit depth, right? So that image was 8-bit, which gives us 0 to 256. So what is a bit? 1 bit equals to 2. Okay, so that's as simple as black and white. As we increase the bit or the bits, what we get is increase in the dynamic range and the gray values that we are looking at. So a 2-bit image has got 4 different values, right? So, and that's based on 2 to the power of n, which is to the power of 2. If we go with 5 bits, that's 2 to the power of 5, which means 32 different shades of grays. So 0, white, and shades of grays, everything in between. Typically, in terms of images, when you are acquiring them on microscope, you will have 3 different options, either 8-bit, 12-bit, or 16 bits. When you are on confocal microscopes, you will be either getting an 8-bit or a 12-bit image. When you are on a microscope with a detector, for example, camera, your options will be 8-bit or a 16-bit, okay? Now, when we compare 8-bit and 16-bit, those images look, or those dynamic ranges look very much similar to you. And that's because the screen that we are looking these images on are 8-bit in nature, okay? Hence, the shades of grays look very similar to you. However, when we look at the numbers, right? 8 bits, they have a dynamic range between 0 to 256, and the pixel values from 0 to 256. If you look at 16-bit, the pixel values range between 0 all the way to 65,000. That's because 2 to the power of 16 is almost 65,500, okay? So once we are clear with dynamic range, we can really go back and try to understand one more thing, which is the image histogram, okay? Image histogram, and what you are seeing in gray over here is log scale of the same thing, which is not in log scale, which is in black over here. So if we look at this image histogram, this is a 16-bit image, right? So boom, now you know a 16-bit image should range from 0 to up to 65,000. And that's exactly what you are looking. You are looking at some of these intensities, which are really, really high over here, and everything less than that, right? So I talked something about saturation. When you are trying to set your imaging parameters, you want to make sure that your image is not saturated, and looking at the histogram really helps you in deciding whether your image is saturated or not, okay? So in this case, we have some of the pixels that are near saturation. And how do we know? Well, we can use a different lookup table. And if we use the high-low lookup table, we know that this particular set of mitochondria are saturated compared to others. So if we break that into individual cells, for example, if I look at the histogram from this individual cell, I know that this is not saturated because values are less than 65,000, they're about 47,000, right? And if I look at this cell now, this is pretty much saturated. So again, rehashing everything, it's very important to make sure that you plan your experiment in advance. When you are on the microscope, you make sure that you're not saturating your image when you are using a particular laser power or particular high voltage or so on. And at the end, a good image will help you get a good data and use the tools in an appropriate manner, okay? So with this, I'll wrap up today's lecture. We will be meeting next week. I'll be posting all the raw data sets that we'll be working on the GitHub page or the GitHub folder. You can get those images out. And the way we will be doing from next week is I will be showing you something for 5 minutes, and you'll be working on that for 5 minutes and go on. So if you have never used Fiji, that's okay. You will not feel strange at all. If you have never done this before, this is totally fine. We will be doing this slow. And our goal is to make sure everybody with all sorts of skill sets feel welcome during our course. Okay. And apart from that, if you have questions while we are going through, let us know. If you think that we are going a bit fast, let us know. We want to make sure that you think about bold experiments in future, right? Sometimes an impediment to you doing a bigger experiment is not knowing how to dupe something. Okay, so if somebody asks me, go and do single cell RNA-seq, I'm like, my God, that's really difficult. I don't know what to do. But if somebody holds your hand and walks you through that, your life becomes easy. And we are here to hold your hand and take you to the next step. Okay, so we welcome you all again to this course, and we look forward to taking you to the next step of your professional career. So any questions, let us know.